Good afternoon, everyone. My name is Lena Fasching. I'm from TU Delft in the Netherlands. And I am happy to be able to talk to you about how we use single molecule techniques to decode aptamer-protein binding kinetics and how that can help us to rational design of aptasensors for continuous protein biosensing. So why is continuous biosensing interesting for us? My PhD is about developing wearable technology to monitor dynamic biochemical processes in the body. So actually what we want to do is we want to target proteins in non-invasively accessible biofluids such as sweat, and thereby provide a tool that allows us for data-driven health management. So that would be, for example, early intervention, or for example, monitoring your health when you're exposed to air pollution, these sorts of things. So when we were approaching this topic, or actually when I started my PhD, I quickly found aptamers because aptamers are actually a very interesting bioreceptor in such a biosensor, because not only have they high affinity, selectivity, I mean, who am I talking to now? But they also show dynamic binding. So actually, they also allow for, or in theory, allow for in situ regeneration. But then we looked for aptamers in the literature. We found some, they had great KD values, and then we actually produced those biosensors, and they were underperforming in our actual biosensing environment. So then we teamed up with some SM-FRET experts in at our university with the CHO lab, and we wanted to study more in depth how these aptamers actually interact with proteins. And why do we want to use SM-FRET, or single molecule first resonance energy transfer? Why do we want to use it? So actually, just as a quick reminder, I assume most of you know the technique. It basically exploits the fact that when you have a pair of fluorophores, if you excite the donor molecule, energy can be transferred to the acceptor molecule, and that emits then again light. And this energy transfer is distance dependent. So only when the molecules are very close to each other, you can really see that. And or you can actually observe that energy transfer. And how we used that in an assay was actually we took the protein of interest, or first exemplary protein, basically. Then we labeled it with the acceptor molecule, and we had the aptamer in solution with the donor molecule. And now when the two molecules are far apart from each other, the fluorophores in the case, then you actually have no intensity, because you also use total internal reflection microscopy, so you only see what is really very at the surface. But when the two come together, you suddenly see an increase in intensity in the red and the green light. And then you can actually really follow single molecule interaction. And what is so cool about it is that you can actually see that in real time. And then you also get the FRET efficiency, so basically a value that tells you something about how much energy was transferred, and that you can then calculate back into the distance. And what we did as a first study is we were looking into literature again, and then we found thrombin, which is like, I think we also heard it today. It's a very well studied example for aptamers in general, I think. So we used thrombin, we mobilized it via neutravidine biotin conjugation on a quartz surface. And then what we were actually seeing, or what our collaborators developed, is a site-specific labeling strategy that uses click chemistry, that 2-PCA-DCBO chemistry, where you can label specifically the N-terminus of the protein. And what that gives you is basically a reference point. So as soon as your aptamer approaches the thrombin, you will only see one FRET energy, or that was at least the hope. So when you had assembled your slide, you could flush through the aptamer solution, and then you put the quartz like under the microscope. And then we saw this. So it's the same field of view. You have the green intensity basically and the red intensity. And what you see is a lot of blinking. And each of these blinking events is actually one binding event. And we were a bit surprised that it's so dynamic because, I mean, we assumed that it would be dynamic, but we were kind of surprised. So then you can actually, with image analysis, you can then get these time traces. So you have a time, over time, you see, okay, each of the thrombin molecules here interacts with the aptamer that is in the solution, and then you have these FRET events, and then you can directly observe how the binding is happening, actually. And you get FRET efficiency. And in our case, this was very constant at 0.4. So with the pair that we used, which was Cy 3 and Cy 5, that translates to approximately 5 nanometers. And that was a good start. So then we had these 3 aptamers that we wanted to test and see how different their kinetic profiles actually are. And then, so we used HD1, which is also known as TBA, which is mainly the G-quadruplex structure. Then we had RE31, which also has this quadruplex structure, but also an extended duplex. And we used NU172, which also has this G-quadruplex structure and the duplex, but it also has some point mutations in the G-quadruplex and also the last loop. And we labeled them all with Cy 3, and then we did exactly that experiment. And what we see is actually that the kinetic profiles vary a lot. So what we actually did is we evaluated the binding and the unbinding time. And then if you do an analysis or include basically more thrombin molecules on your surface that you mobilize to get these binding and unbinding times. And there you can already see that HD1 shows the lowest or the shortest binding time with only 1.5 seconds, while NU172, so here has the lowest, the shortest binding time, while NU172 shows actually the highest binding time with almost 10 times as much. And RE31 is with 3 seconds. The unbinding time, so that's basically the frequency of binding events, was the highest for RE31 and the lowest for NU172. So then you can, from these unbinding and binding times, calculate the K on, K off, and KD values, which you can then compare with literature. And what we found is actually interesting, or at least it was interesting to us, that you have very different kinetic profiles, but you end up basically with a very similar KD value. So what this technique allowed us is actually to observe this very transient binding behavior and get a real-time kinetics, so to say, of the individual with these individual binding events. Now then we asked ourselves the question, okay, how can we then use this information that we got from the SM FRET into the binding biosensor, because that was ultimately our goal. Now, and that is something that we want to stress, the selection of aptamers is really dependent on the biosensing context. So for us, we wanted to use proteins. We also wanted to use, we also want to detect proteins in sweat. So we actually assume a very low protein content in sweat. And we also need some sort of continuous flow in our sensor, because otherwise you're always measuring the same sweat. So you don't have really new information. So yeah, that's what were our assumptions. And then that led us to thinking that we were actually not really reach an equilibrium in our sweat sensor, but we will most probably measure this association within the pre-equilibrium state. So we wanted to actually see if these assumptions are true, and what does that tell us about which aptamer we then have to select. And we used the QCM, because the QCM is a very nice technique where you can actually see a response of your aptamer in real time as well. So what is the QCM? It's called quartz crystal microbalance. It's based on a quartz that is a piezoelectric material. You have a gold electrode on the top and on the bottom. You connect the electrodes to alternating potential. So you apply alternating potential, and you let this quartz oscillate at its resonance frequency. And then this frequency will change as soon as there is mass added to its surface. So it's a mass-dependent transducer that will also give you a response here in real time. And what you can also do, and that is also one reason why I chose, is you can operate it under flow conditions. So you can actually really simulate this continuous biosensing example. So you have an inlet and outlet, and then you flush through your target solution. So we did a quick computational simulation just to show how thrombin, or just to see how thrombin is distributed in your sensing chamber. And then you can actually start to think about, okay, which parameters are going to influence our sensor response. So flow rate was the first thing we simulated, and we saw, okay, with increasing flow rate, we see an increase in sensor response. So this represents here this drop in frequency would be a higher, lower drop to a lower frequency actually means higher sensor response. So we see that with increasing flow rate, we see an increase in sensor response, and that actually led us to the assumption that we really are in this mass transport limited system. And then we continued to check, okay, is this thrombin concentration dependent? Our sensor response was kind of a sanity check, and we really saw, okay, we can use it also in this association phase as a biosensing example. And then we actually checked, okay, how do our kinetics influence our sensing example? And we saw, okay, the K-on rate in this mass transport limited example does not really have as much of an influence as the K-off rate has. So here you can see we varied the K-off rate here with 0.01, between 0.01 and 1. And what you can actually see is that the longer the binding time, the higher your sensor response. And that was actually very interesting, because then we actually wanted to see, okay, which aptamer is now the better one. So we did it in the lab. We coupled our aptamer through EDC and HS chemistry and the use of a self-assembled monolayer to our surface. And then we checked the two aptamers that showed a very similar KD value, so RE31 and NU172. And remember, they had this very different kinetic profile. So NU172 had a lower K-off rate, but RE31 had a higher frequency, so the higher amount of binding events. And we actually see that what we found was actually very was in correlation with aligned with our computational simulation, because we can see that here the frequency dropped more for NU172 in comparison to RE31. And then when you do the calibration curve, you can also see, okay, there's a linear correlation, and for NU172 it was steeper. So it really helped us that despite the similar KD values we found for the two, we could select the one that in our biosensing context gave the better response. So the key takeaways is actually we wanted to advertise here SM-FRED as an interesting tool to characterize aptamer target interaction in real time and at the single molecule level. We can also see with these methods the subtle differences in the binding mechanisms of aptamer, because we can really visualize this distribution in K-on and K-off values rates. And then, we show you basically that the KD value is not always a good value to take when it comes to aptamer selection for biosensing. And then looking forward, we are actually trying to reverse the scheme, because you saw that we first immobilized the thrombin, then we immobilized the aptamer. So we hope that by reversing the scheme, we can enhance the translation for because then we also want to go to different aptamer target pairs. So in the end, I just want to thank my collaborators and my PI for their guidance, and also you for listening.